Hello students, hope you all are safe. The COVID-19 pandemic and lockdown should not stop us from learning. In today's session, I will be introducing you all to the concepts in genetics, which is discussed in the chapter Inheritance and Variation. For your reference, please ensure that you download the PDF of the biology textbook from the Maharashtra State Board textbook download website. Continuity of life and perpetuation of species is due to a remarkable characteristics of living organisms to reproduce. All organisms inherit certain characters, which are also called as traits from their parents and in turn transfer these traits to their offsprings. Therefore, offsprings resemble their parents and they resemble each other also. But have you ever noticed that they are never exactly identical to their parents? At times, they also show certain differences. Every living organism possesses characteristics of a particular species, but can be identified from each other of the same species due to certain differences. Similarities and differences are the two aspects of the same mechanism called as inheritance. So, before starting this chapter, we need to understand certain terms. The first important term is heredity. It is transmission of characters from one generation to the next or from parents to offsprings. Heredity is simply means the characters being transferred from one generation to another. At the same time, we have variations. It is the differences between the parents and the offsprings, among the offsprings of the same species and among the individual of the same species. Have a look at this image here. It's a picture of a family. You can see that from individuals from one generation to the another, though they belong to the same family, they are showing a number of differences. They are not identical to one another. And genetics, it's a branch of biology that deals with heredities and variation. Students, the first scientific explanation regarding inheritance was given by Gregor John Mendel in 1866. He performed experiments on garden pea in a very scientific manner and proposed postulates or principles which are now popularly known as Mendel's laws of inheritance, and his epoch-making work is known as Mendelism. He laid down the foundation of genetics and therefore he is rightly called as father of genetics. Let me highlight some of the works that he did. Gregor Mendel conducted hybridization experiments on garden pea for 7 years and proposed the laws of inheritance in living organisms. During Mendel's investigation into inheritance pattern, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiments had a large sample size, which gave greater credibility to the data that he collected. Mendel's investigated characteristics in garden pea plant that were manifested as two opposite traits. Traits means the characters that you can see, the features of an organism. For example, in his case, it was tall or dwarf plant or yellow or green seeds. Based on these observations, Mendel's proposed that something was being stably passed down unchanged from parents to offsprings through the gametes over successive generation. He called these things as factors. Now we call them as genes. Therefore, genes are the units of inheritance. Mendel's was a genius, much ahead of time, as he gave these concepts of factor before the discovery of mitosis, meiosis, chromosomes, and without the use of microscope. Unfortunately, his work remained unnoticed. He died in 1884 as a highly respected priest and teacher, but unrecognized as a scientist. In 1900, his findings were rediscovered by three scientists, Hugh de Wern's, Carl Corin, and Erich Schomack. Independently, they found Mendel's work to be very significant. During these years, mitosis and meiosis were discovered, and the scientific world realized the greatness of Mendel and his work. Based on Mendel's principles, that is his findings, Corin postulated three laws that are known as Mendel's laws of inheritance, and a new branch in genetics in the field of biological sciences was opened. Students, it's very important to highlight the reasons for Mendel's success. Let me tell you some of the reasons why he was very successful in his experiments. His experiments were carefully planned and involved a very large sample size. He carefully recorded the number of plants of each type and expressed his results as ratios. In the pea plant, contrasting characters can be easily recognized. He was lucky because the characters that he used were always having one a trait at a time and a contrasting trait with it. For example, if he was working with a tall plant, the contrasting character that he had was a dwarf plant. He never came across any intermediate characters. The 7 different characters in pea plants were controlled by a single factor each. The factors are located on separate chromosomes, and these factors are transmitted from one generation to another. Factors, let me mention, is the term that Mendel's used for that particular particle of our genes which was responsible for transmission of the characters. He was the one to introduce the concepts of dominant and recessiveness. Students, in order to understand Mendel's experiment, we need to travel back in time to understand what was in Mendel's mind before he started his very popular hybridization experiments. There were a few questions which came to his mind. How are the characteristics transmitted from parents to offsprings? Why are there similarities as well as differences? How certain traits reappear in grandchildren? The answers to all these questions were to be found out through experimentation. Now, can you think about a suitable experimental material for the hybridization experiments? What characteristics of the experimental materials are expected? Now, the success story of Mendel was because of his selection of his experimental material, that is the garden pea, which is also called as Piscium sativum. Garden pea is an annual plant and completes the life cycle within 3 or 4 months. Due to this short lifespan, he could grow 3 generations in a year. It's a small herbaceous plant that produces many seeds, and so he could grow thousands of these seeds in a very small plot behind his church. It was a self-pollinating plant and was available in forms of many varieties with contrasting characters such as tall and dwarfness, yellow and green seeds, violet and white flowers. There were no intermediate characters. The flower was large enough for easy emasculations for artificial crosses. Emasculation is a process where the anther is removed well before anthracis, that is the development of pollen grains. With this help, you could convert the flower into a female flower. Now, this plant was also known to produce fertile offsprings. This image alongside shows Mendel's 7 pairs of contrasting traits. For example, the flower color, that is purple and white, seed shape, round, wrinkled, seed color, yellow, green, the pod color, green, yellow, shape, inflattened, constricted, the height of the plant, tall, short, the position of the flower, axial, terminal. These were the different traits that he used. He's also mentioned the trait that was dominant and recessive. Thank you. you students for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe and keep learning.